Good morning, students. Welcome back after the winter vacations. I hope all of you had a good vacation. You were able to relax. You were safe and your family was safe. Now, school has started and we will also start with the online classes from today. I will be teaching cell cycle and cell division. Today we will start with this topic. Now in class 10 you have studied about cell division and cell cycle and you have a brief idea of that. So the first lecture is almost going to be a revision of what you have done in class 10. Thereafter, in the next class, I will start with the second kind of cell division, that is meiosis in detail. You have already studied about mitosis and its stages in detail in class 10. So today I will be revising that topic, and as I have told you in class that you must read this topic in detail so that you will be able to understand meiosis later on. So let us begin. Now you know that all cells have a basic lifestyle. Rather, I would say all cells go through a life cycle, and this is a very important process for all living organisms. The cell is the basic unit of life, and scientists like Sheldon, Schwann and Rudolf Virchow have given the cell theory, and according to it, cell is the basic structural and functional unit of life, and all cells come from pre-existing cells, as told by Rudolf Virchow. A cell cannot live forever. The continuity of life depends on cell division. And you know that there are two types of cell division. One is called mitosis, the second one is called meiosis. And two major events occur both in mitosis and meiosis. These are the first one is called karyokinesis, which means division of the nucleus. And the second event that follows karyokinesis is cytokinesis, that is division of the cytoplasm. Now, mitosis retains the chromosome number. You know, each species has a specific chromosome number which is fixed for a particular species. For example, in humans, the chromosome number is 46. So 46 chromosomes are found in each cell. And when I say mitosis retains the same number of chromosomes in daughter cells, it means that the new cells that are formed from the parent cell, that is the daughter cells, will also have the same number of chromosomes. And this results in the multiplication of cells. Whereas in meiosis, the other kind of cell division, the chromosome number becomes half in the daughter cells. And this kind of cell division, that is meiosis, occurs during the formation of gametes. So this was taught to you in class 10. So that is why I was saying that what I will tell you today will be revision of what you have done in class 10. Now we come to cell cycle. All cells go through a basic life cycle. And as I said, it is a very important process for all living organisms. Now, before the cell divides, cell growth and replication of DNA. Replication means duplication of DNA will occur. And these three processes, that is growth of the cell, DNA replication, which will then be followed by cell division, occur in a very sequential, coordinated manner. If we have to define cell cycle, we can say cell cycle is the sequence of events by which a cell duplicates its genome, then synthesizes the other constituents of the cell, and finally divides to form two daughter cells. Now, cell growth is a continuous process, but synthesis of DNA, that is replication of DNA, occurs only once in a cell cycle. Now, this whole sequence that occurs in a cell cycle has been divided in different phases. And the duration of the cell cycle will differ from organism to organism and also from one kind of cell to another kind, or we can say from cell type to cell type. And the cells may vary in the amount of time also that they spend in the different stages. Like I will give you an example. Bacteria like E. coli divide once in every 20 minutes, whereas the epithelial cells divide every 8 to 10 minutes. And the cells found in the root tip of onion, they divide every 20 hours. And therefore we say that the amount of time that is spent by the cell in different stages of the cell cycle will differ. But basically there are three main phases in a cell cycle, and these are interphase, M phase and cytokinesis. Interphase is the resting phase. This is a very active phase. Though we say this is a resting phase, because cell division has not started as yet in the interphase, but it is a very active phase in the cell cycle because DNA gets duplicated over here. And the second phase, the M phase, is the mitotic phase in which the cell division takes place, and the third phase is cytokinesis, in which division of cytoplasm takes place. Now we will discuss each of these phases one by one. For example, we begin with the first phase, interphase. As I just told you, interphase is a very active phase, and DNA is duplicated here. The cell actually appears to be in the resting phase, but physiologically it is the most active stage. It is this time during which the cell prepares for division by undergoing both growth of cell and replication of DNA in an orderly manner. Now, this figure actually shows you the cell cycle, the different phases of interphase, interphase is also divided into 3 phases, as you can see in this figure. G1 phase, the S phase, S stands for synthesis phase, and the G2 phase. G1 and G2 phase are the growth phases, and S phase is the synthesis phase because synthesis of DNA occurs in the S phase. Then comes the M phase. M phase is karyokinesis followed by cytokinesis. That is, the division of the nucleus will take place, and there are 4 sub-stages of division of the nucleus, as you can see in the figure. Prophase followed by metaphase, then anaphase and telophase. And here, after these 4 phases of division of the nucleus divides to form 2 nuclei, and then the division of the cytoplasm takes place. So we have 2 daughter cells at the end of the cell cycle. So this is a general figure depicting the diagrammatic representation of the cell cycle. Now, as I was discussing the interphase, and I told you, physiologically, interphase is the most active phase. Now we will be doing the 3 subparts of the or subphases of the interphase. The first phase is called G1 phase, or we also call it the gap 1 phase. And this G1 phase corresponds to the interval between the mitosis and initiation of DNA replication, because DNA replication will take place in the S phase. So G1 phase is the interval between mitosis and synthesis of DNA. The cell is metabolically active and it continuously grows but does not replicate its DNA in this phase. The major events that take place are as follows. Cell will grow, mitochondria, chloroplast and lysosomes, endoplasmic reticulum, Golgi complex, vacuoles and vesicles will be produced. Structural and functional proteins will be formed. Nucleolus produces ribosomal RNA, messenger RNA and transfer RNA. Then ribosomes are synthesized and the metabolic rate of the cell becomes very high. These are the list of events which I have mentioned point-wise just now during the G1 or the gap 1 phase. The gap 1 phase is followed by the synthesis phase. And synthesis phase actually marks the period when DNA synthesis or replication or duplication of DNA occurs. That is, to explain this, if I say initially a cell has 2n number of chromosomes which can be denoted as 2C, the amount of DNA can be denoted as 2C, then it increases, it becomes double, it becomes 4C. But the number of chromosomes does not change. The number of chromosomes remains 2n only. That is the diploid number of chromosomes. There is no increase in the chromosome number, but the DNA becomes double, that is DNA becomes 4C from 2C. And after S phase, the number of chromosome is going to be same as diploid number, that is 2n. The major events that take place here are first, as I told you, replication of DNA will take place. Besides this, protein molecules, that is the histones, are synthesized which will cover the DNA. And each chromosome is in the form of 2 chromatids. The centrioles, which help in cell division, they will also replicate in the S phase. Then S phase is followed by the G2 phase. And G2 phase, as I told you, is gap2 phase. The major events here are proteins synthesized in the preparation for the mitotic phase, that is the division phase. The spindle begins to form, the mitotic spindle begins to form, and store of energy increases. There is increased metabolic rate and cell growth continues. Now, during the cycle, growth occurs throughout G1 and G2 phases. And these phases separate the synthesis phase from the mitotic phase. But some cells in adult animals do not exhibit cell division. That is, cells of the heart, the muscle cells, the cells of the nervous system, brain cells, that is the nerve cells or the neurons, these do not undergo division. And there are many other cells which divide only occasionally when they are needed to replace the worn out cells and or cells that are lost because of some kind of injury or death of the cell. So such cells do not divide further, and they enter an inactive phase which is called the G0 phase, or we even call it the quiescent phase. G0 or the quiescent phase is when cells that do not divide, they exit the G1 phase and enter into the inactive phase called the G0 phase. Cells in this state remain active metabolically, but they do not divide unless required. And I will give you an example here that the liver cells, they do not divide, but they can resume division and enter the G1 phase in response to factors which are released during injury. Liver cells can divide, but that is under extreme cases of injury to the liver. And feedback from the cell determines whether the cell cycle will proceed to the synthesis phase, whether it will pause for a short while only or withdraw into G0 phase for a long extended rest period. Then comes the M phase, which is also called the mitotic phase. Now, M phase represents the phase when division of nuclear material takes place, and this is called karyokinesis. There is equal division of DNA in the daughter cells, the cells that will be formed, and M phase takes only a small fraction of the entire cycle. Nuclear division, as I told you, occurs in 4 main stages: prophase, metaphase, then anaphase and telophase. After this, I will show you the diagrams of the 4 phases of karyokinesis, which you have studied in detail in class 10. So I will not teach that. So you have to revise that on your own. Then the next phase, the last phase after M phase, after division of the karyokinesis, is the cytokinesis. Or here the cytoplasm of the cell will divide and it is referred as division of the cytoplasm, cytoplasmic division, or cytokinesis. And the cell membrane divides, daughter cells are formed, and these daughter cells, this is a point to be noted, are identical, exact copies of the parent cell. They are identical in all respects. There is an equal distribution of cytoplasm and even all the organelles into each daughter cell. So this was cytokinesis. Now, as I told you, cell division is of two kinds, mitosis and meiosis. Now, mitosis is the cell division that occurs in the cells of the body. We call them the somatic cells. The cells that are formed are identical in all respects, having the same genetic constitution. Now, I have listed some points about mitosis here. You go through them, and these are the different stages depicted by diagrams. You can see them, but the major events you have studied, so I am not teaching them here. This is prophase. This is the longest phase, and in prophase, as you can see, that the nucleus, the chromatin network of the nucleus has changed into chromosomes. The centrioles have started dividing. The nuclear membrane has started disappearing, and the microtubules radiate from the centrioles, which you can see in the next stage, that is metaphase. There is complete disintegration of the nuclear membrane. The chromosomes are spread in the cytoplasm of the cell. There is formation of spindle fibers, and this stage, the chromatids of the chromosomes, they line up at the equator. This is an important feature of this phase, that the chromatids line up at the equator, and the characteristic feature of metaphase. This figure shows you a detailed structure of the chromosome with the centromere or the kinetochore. Then after metaphase comes the next stage, the anaphase, and in anaphase, as you can see, that the chromosome splits at the centromere, and the daughter chromatids are then pulled towards the poles by the shortening of the spindle fibers, and each of these chromatids are now called chromosomes. So you can see that initially there were 4 chromosomes. Now after splitting of the chromosomes, 4 move towards one pole and the other 4 move towards the other pole, and each pole will have one set of chromatids. And after anaphase comes the telophase. Telophase is actually reversal of prophase. The events that had taken in the prophase are reversed, like nuclear membrane will be formed, the chromosomes will uncoil, they will lengthen to form the chromatin network. The spindle fiber that was formed in the prophase will now disintegrate, and each part of each pole will have a centriole. The nuclear membrane will reappear, and at the end 2 nuclei are formed. And then you can see by the depression here that the cell membrane forms a furrow, and there is division of cytoplasm, that is cytokinesis will occur, and the cytoplasm will be divided into 2 equal parts after the nuclear division, and 2 daughter cells are formed. So these with the help of these diagrams, I have explained in short mitosis, but you read about mitosis and make your own notes in detail. Then we come to the importance of mitosis, that is, the significance of mitosis. Now, mitosis basically has led to the formation of two daughter cells with identical gene composition. The cells contain the same number of chromosomes as the parent cells. The cells carry the same hereditary information as in the parent cell. There are no variations in genetic information. There is genetic stability within a population. Mitosis leads to growth, that is, the number of cells increase and there is growth in the body and there is scope for cell differentiation. Then mitosis leads to cell replacement and repair, that is, the lost cells of the skin, the blood cells, the cells of the digestive tract, and wounds that have been caused will occur, wounds will be healed, and the cells of the upper layer of the epidermis form the lining of gut, and all the cells of the blood are also replaced. Then another important importance of mitosis is regeneration and asexual reproduction. Now, by regeneration, it means that if a part of an animal is lost by cell division, which is a kind of multiplication, that lost part will be regenerated, and it is seen in animals like planaria and sponges. Whereas in case of plants, asexual reproduction occurs, which is a kind of vegetative reproduction, and the number of the species increases. Then mitotic divisions in plants in the meristematic regions, like in the apical meristem, terminal meristem and lateral cambium, results in a continuous growth of plants throughout their life. Sometimes what happens that because of uncontrolled divisions, mitotic divisions, tumors are formed, and which these tumors can also be cancerous in nature. So this was about mitosis and the various stages of mitosis, and the most important thing is that you have studied about cell cycle in detail with its phases, like the G1 phase, S phase and the G2 phase, and the events that take place. So this much is enough for today. Revise, make your own notes, and be serious now, because you have limited time in hand and a lot to do. So good luck to all of you. Take care, stay safe and stay healthy.